Hey, good morning to everybody who's joining us live, and if you're watching this recorded, good hello to you. All right, we're going to go ahead and talk a little bit more about membranes like we have been doing last week in class. So some of this will probably be familiar to you. So the first part is probably a review, but then we'll get into it probably a little bit deeper than you have in other biology classes. So, if you think about transport, we're going to kind of focus on energy here in this first part. So, in this little picture here, you've got a membrane, you've got certain things crossing. You can see there's different concentrations on each side. Those are things that should probably be jogging your memory about energy and transport. So just as an introduction, there's several ways that cells can use transport to maintain homeostasis, especially for water and other solutes. So we put these two things right up here into two categories: passive transport, no energy, and active transport, which requires energy. So let's take a look at this. Are those sounding familiar to you? You can give me a yes in the chat if you've heard of this stuff before, or no in the chat. Let me check that real quick. Familiar? Thank you. Okay. See a lot of yeses coming in. So just this first part will probably be a review. So we have two sort of beakers here. This is a before and after. This is not things moving over. So when we look at sort of like our key terms, they'll be over here on the side. Those are things you definitely need to understand. Passive transport is also called diffusion because it doesn't require any input of energy. Molecules spread out. They have this natural motion. I think it's called Brownian motion. And that, if they're sort of spread, or if they're sort of arranged together, they will spread out to an equal concentration throughout the container. So if this was a drop of, say, I don't know, some substance we put in here, it should and it will spread out throughout the container. Now, this doesn't happen instantaneously. This does take a little bit of time. There are some sort of things you have to keep in mind when we look at passive transport and how it works. And those are things like concentration. Is it saturated? But in general, it's pretty simple because diffusion makes it go from a concentrated area to sort of a spread out even situation like we have here. Okay, let's move on. So on a cellular level, this is happening on usually carrier proteins. In our last lecture, we looked at some substances that could pass through cell membranes, and those were small non-polar molecules. So water can go through it very slowly, but if you want something to go in faster, you usually have these proteins, carrier proteins, transport proteins on the membrane of the cell that will either open or be open to particular substances. So usually these are selective. They're only going to let certain things through. Okay. Moving on here. So this is just to review, this is just kind of what I was talking about. Small non-polar things. The big thing to sort of notice here is, I'll try to draw a transport protein. There are no transport proteins in this section here because we're talking about things actually going through that phospholipid bilayer. And this can go both ways. It can go in and out depending on, especially for osmosis, depending on the concentration of things that are inside and outside the cell. If you have a high concentration of, say, salt or sugar or glucose or whatever, water will try to move outward to balance that concentration of glucose on whatever side of the membrane we're talking about. So new section here. We just went from active transport, we just went from passive transport. Now we're talking about active. Active transport requires energy in the form of ATP. ATP will cause these transport proteins to change their shape. So this transport protein right here will have a binding site for these triangle shaped, this one's passive. This one doesn't use energy. These will just pass through here. Let me go back to this one down here. This active transport protein will have a binding site somewhere on its surface or opening for these square shaped particles. And the concentration out here might be very low, so we're going to concentrate these so it's the opposite of passive, and that requires energy. Once these things bond with this transport protein, that transport protein sort of traps them and opens up a one-way door that will spit them into the inside or outside of the cell, depending on which way it's going. We're going to talk more about that in 2.9. So the big thing for active transport is you're going from low to high, and it does take energy, and that energy is from ATP. So this is probably all review, right? Okay, I see some heads nodding up in the camera section, and I appreciate you guys showing me your faces a little on camera so I can see those things like nods and thumbs ups. So that was a really quick section. So you have to be able to define active and passive, compare and contrast. Compare and contrast, energy for active, high to low for passive, low to high for active. So pretty easy stuff, a review for most of you. All right, let's go to a new section here, endo and exocytosis. Talk about those for a sec. Okay, some things are too large to go through the plasma membrane, so there are other things cells can do to get those in and out. Some things are also too large to go through transport proteins. So transport proteins can have sort of like limits to their size. If you want these things to get into the cell, you have to use another way. So as we said in the last one, the structure of that cell membrane gives concentration gradients, and that includes, in this case, large molecules like these G's here. So let's take a look at what happens with larger molecules. What we have are this is where the organelle vesicles come into play, and vesicles can be used to transport things within the cell without mixing. So if you want say you have something in here, some sort of particles, whatever's in here is going to stay in here and not mix with what's in the cell. Now, what these things do, these are phospholipid bilayers, so these are internal membrane-bound organelles. When these bump up against the actual cell membrane, they can sort of burp out their contents, okay? So just a review of what a vesicle is. So this can work both ways, okay? So you can have cells, this kind of shows it going in and out. So taking things in, just kind of keep these clear here. Taking things in is endo, and out or excreting is exo. And I always remember exo is out, and that makes the other one pretty easy to sort of keep in your memory. So what's going on here? The cell membrane will make this sort of movement, and they call this invaginating, and that is related to the word that you can probably hear in there. It makes a compartment, and so this invagination will surround food particles, and then the membrane will pinch off, so you can kind of see this in sequence here, and whatever is in here is separated from all the other areas of the cell. In animal cells, lysosomes will merge with that membrane, and there's digestive enzymes in here. They're going to remove the useful products, and then the opposite happens. This vacuole is now full of digested material. Whatever the cell doesn't need merges with the membrane and kind of burps this out. So over here, think of this as a mouth. So if I kind of make this a cartoon, the cell is sort of eating here, my cartoon head, and then it's going through the digestive system and sort of popping out on the back end. It doesn't always happen in a straight line like this, but for teaching purposes, this diagram's pretty simple, okay? So this is an example of it happen. There's other ways it happens, but this is sort of the most clear-cut, easy to understand example. All right, let me erase those drawings and move on to my next one. We're got a few more to go. This is not a terribly long lecture. Okay, exocytosis also happens, and focus on exit there, for things the cell needs to transport, usually into an organism, okay? So this is where we go back to organelles again. Whatever protein that this is manufacturing, if it's needed outside the cell, then this protein will get produced in the ER, get packaged by the Golgi, and those membranes that this thing is forming kind of pinch off from the Golgi. You can see all these little things that look like bowls or coconuts. Now remember, those are fully surrounded. They're cutting those in half so students can see what are inside of them. And these ones will merge with the membrane and secrete whatever protein is needed by the organism. And here is sort of a close-up of the membranes merging, okay? So this is whatever needs to be secreted, and the membrane of this area right here merges with the external, sort of the main cellular membrane. So this is an internal membrane. When this sort of bumps up against here, it merges and it either pulls things in or lets things out depending on which way we're going. And that's because that membrane is constantly moving. Don't think of this like a wall. Think of it like a soap bubble or something like that. Like soap bubbles can combine, and that's sort of like a macro example of this happening. It's not exactly the same, but it's similar. All right, everybody okay out there in the Google Meet? Pacing okay? Drawings okay? Give me a chat there. Oh, Brian has a question here. Is phagocytosis just an umbrella term for endo and exo? I think phago is eating, so I don't think it includes exo. I think phagocytosis is an endo process only. Good question. Okay. But here's an exact example of that. So because the membrane is so fluid, it's able to sort of pinch around. And remember, I went over this already. Um, in endo, it does take energy to move the membrane this way. So you're looking at the cytoskeleton and other motor proteins bending the membrane into this pocket right here. So invagination does take energy. So we process both, because they use ATP to deform the membrane and make these pockets. These are active transport. So endo and exocytosis are active. So this should answer the question that was just asked in the chat. There's categories of this, okay? Phagocytosis. Phago means to eat. So some sort of pockets will be formed on the cell membrane, and that could be either pseudopods or invagination, like we said, and they are going to pull that in and pinch off. And this is kind of a new term here. A phagosome is a membrane that is created in phagocytosis. Okay, pinocytosis is really specific to water and other liquids. So same process. Remember, these are all forms of endo. So endo divides out into these three types. So pino is any kind of liquid. Next we have receptor mediated. There'll be receptors on the cell. Those will bind with some extracellular substance, and when that happens, the membrane will again fold in and pinch off. So 3 types of endocytosis you should be familiar with. Questions? Can everybody okay out there? All right. So just another cartoon that shows this. So exo is going out, endo going in. Okay, so exit for endo. Or I'm sorry, exocytosis for exit. Said that backwards. Almost done here. Just going to talk about some review. So these terms here, this was defined in the lecture before this. This is 2.6, but you still got to know how selective permeability applies to these situations. Tell me what a concentration gradient is. There are more particles on one side than the other, so it's going to be more concentrated on, you have to be able to say where it's more concentrated. Talk to me about how membrane fluidity with phospholipid bilayers plays a role in the transport of these large molecules. Well, you can say to me, the membrane will fold around and merge with the cellular membrane to allow endo and exocytosis to happen, which leads right into this. You're going to tell me the difference between those, the 3 different types of endo, which is your final goal right here.